Microarrays are used to get the idea about the gene expression, and we see that there are two main types of microarrays. One of them is slide-based arrays, and the other one is Informatics, where we use specially designed chips. Slide-based arrays, the probe is fixed directly to the surface of a glass microscopic slide, so the probes are there on the slide. And these probes, they can range from a size like maybe 60 nucleotides, or we can also have entire long cDNA which is present over there on those slides. So we take, for example, here again we repeat this example. We have seen that earlier also. So we have RNA type 1 and we have RNA type 2. We take those RNA type 1 labeled with this CY3, which is a dye, and then we have CY5, which is another dye that is colored red. So we take these two RNAs and we let them hybridize to the probes which are present on the slide. So the slide has the probes, and these two type of RNAs that may be coming from two different tissues or may be coming from the same tissue, but we might have some treatment versus one of them is a control. So we put them together on that slide where we have those a lawn of probes are there. So they will hybridize. We see in some areas we have more red color, so it means this tissue which is actually labeled as CY5, that is relatively more as compared to the green one. We can also observe the green spots in which the relative proportion of the other type of RNA is higher. We can also have these yellow spots where we can have a mixture of both of them. Whereas in Affymetrix, we have actually those small chips. So we have actually those nucleotides, and the probes are actually 25 nucleotides in length. We do have a mismatch pair in between them. I'll explain in the next slide why it is there. There is a single mismatch at the center of the probe. So it is used to detect and eliminate the false results. So actually, where we do have mismatches, just one mismatch, the color is different. But then if there are more than that, we will not tolerate. So in that case, we can remove those spots which are having those false positive results. We do have 11 probes per gene, and these slides are prepared by the process called as photolithography. So here is Affymetrix in detail. So we have these stations where we have thousands of those small squares, sections. We can call them as stations, and then in each station we have those millions of small DNA molecules which are attached over there. Those nucleotides actually which are the probes, so they are attached over there. Here you can see the relative area. You can have a scale in your mind if you want to compare these pictures. And then you have in each region we have those probes. So we see that we have those different probes for a single single RNA species. So we have 11 probes per different species. Here comes our RNA, which is the target RNA. So they come and attach to those probes which are present on those chips. So when they attach on them, they do have some tags which are attached. So those tags are actually then they pass through those. Actually, these are molecules. We call them as phycoerythrin and streptavidin complex. Actually, these target RNA molecules, and they are attached with the biotin, and then biotin is then attached with the streptavidin, and then streptavidin has attached phycoerythrin, which gives the color when laser is passed through that. So after the hybridization, we get those spots which are highlighted by using those colors, and then we can get the relative intensity of those spots. So that is the measure of the expression. So here we see that. So we have those large number of red spots. Red are where we have those hybridizations. So we do have some areas like blues or some other colors which are actually the spots where we did not see any hybridizations. Now this is how it looks in the machine. We have RNA, we convert it into the cDNA, then we attach those biotin to the cRNA. cRNA is actually the antisense from this cDNA, so that is labeled with biotin, so that's why it's called as biotin cRNA, copied RNA. Then this is hybridized to the probes which are present on those chips. In this hybridization, we have discussed earlier that streptavidin and phycoerythrin, and they are also attached. They play important role. They give the signal for the attachment of those RNAs after this hybridization. So this is how you see a colored pattern in it, which are detected by the softwares in the computer. So then they convert it into these signals, which are actually based upon the relative proportion of these colors. So they give the idea about the RNA expression. So what is the difference between slide and Fe matrix? Fe matrix, high density oligonucleotides are there, and in the glass slides we have cDNA, long oligos, etc. They are conceptually similar, but the designs and the manufacture processes are different. This Fe matrix is actually coming from a big company which was also located in California. So they had actually big shares in the market. So they had been in microarray technology. They have recognized their technology as one of the best technology. And since these probes they are making, these chips they are making, they are relatively improved over the different as the time progresses. So that's why their technology was more famous. So microarray are used to investigate gene expression, and we see that there are two main types. One of them is those slide based arrays, and the other one is these Fe matrix.